Thank you for attending my talk. Today, I will present my works on high-dimensional directed network analysis during my PhD research. My research interest is to study the human brain. I want to understand the functional organization of the human brain, how it brings about different brain functions, and how it varies during the neurological or psychiatric disorder. Overall, we view the brain as a high-dimensional directed network system. Each network node represents a brain region. Each network edge represents a directed influence exerted by one region on another. We called it directed connectivity. We also called it as directed functional connectivity or effective connectivity. We want to know how brain regions interact with each other and form different brain network patterns in performing different functions. To answer this question, we develop statistical modeling methods to reveal high-dimensional directed brain networks using brain data. In today's talk, I will present two of my models. The first model is for studying directed brain networks of patients with epilepsy using intracranial EEG data. Intracranial EEG data are multivariate time series recordings of brain activities of patients with epilepsy. And we use this data to understand the pathological directed brain networks of patients with epilepsy. Later in this talk, I will explain more about epilepsy and intracranial EEG data. Our second model is for studying whole brain directed networks using many subjects fMRI data. And in this work, we also develop an efficient method to address the computational challenge of analyzing thousands of subjects' fMRI data. And so now let me first explain my first model, and I will first explain epilepsy. Epilepsy is the fourth most common neurological disorder, affecting approximately 3 million people in the United States. In an epilepsy setting, when the seizure occurs, the abnormal, excessive, overly synchronized neuronal activity starts at a specific brain area called the seizure onset zone and spreads quickly to other brain regions. Now let me show you a video about what a seizure looks like. Let me stop sharing. Let me turn to the video page. Can you see the YouTube page?
Yes, I can see it.
Great. So this is a patient with epilepsy, and this window recorded her behavior, and the background, the time series plots are the recordings of her brain activities at the same time. So now let me start the video. At the beginning of the video, the patient behaved normally, and she was watching her cell phone. Then she started to feel the seizure and called the nurse. We observe abnormal activities in some of her brain regions. As the time passed, we can observe more and more her brain regions show abnormal activity. And this patient started to show abnormal behavior, symptoms. Her muscle jerked, her body shook. Eventually, this patient lost her consciousness, and we can observe that the abnormal activity spread to her entire brain. Now let me go back to slide. Can you see the slide? Okay, thank you. Well, as the video shows, the can seriously affect the life quality of a patient with epilepsy. And typically, patients with epilepsy take medication to control their symptoms. Unfortunately, more than 30% of patients develop drug-resistant epilepsy. So this indicates that the medication either has no effect on controlling their symptoms or gives this patient too many side effects. For these patients, the best chance to achieve seizure control is to surgically resect the seizure onset zone. Therefore, localization of the seizure onset zone is critical to the success of the surgery. To localize the seizure onset zone, neurosurgeons place intracranial EEG electrode directly on the exposed surface of the patient's brain. Here is an example of the intracranial EEG electrode placement. Each white dot is one intracranial EEG electrode. It records the brain activity of a local brain area. And with high temporal resolution, high spatial resolution, high signal-to-noise ratio, all nice properties. Here is an example of the intracranial EEG recording. To localize the seizure onset zone, trained EEG experts visually examine the intracranial EEG data, especially around the seizure onset time. They detect the region that first has abnormal activity at the seizure onset zone. However, brain is such a highly complicated dynamic network system, and the subtle abnormal activity of a seizure onset zone may not be always identified by human eyes. So this visual examination sometimes fails. As statisticians, we want to develop statistical methods to help localize the seizure onset zone. So how should we achieve this goal? As the video shows, seizure propagation is a directed and dynamic network phenomenon. The directed connectivity from the seizure onset zone to other normal regions first changes at the seizure onset time. So detecting this change should be useful for localizing the seizure onset zone. We also conducted literature review and found existing studies also selected that the functional connectivity and directed connectivity are useful in seizure onset zone localization. So we believe that we can improve the localization of seizure onset zone and improve the treatment by reviewing the directed brain network of patients with epilepsy. Now the question is, how should we quantify this directed connectivity? Existing methods for directed connectivity fall into two major categories. One is the information theoretic measure, such as the cross correlation and the transfer entropy. However, such measures focus on pairwise relationships, and they cannot delineate the entire pathway of signal transfer that can transit through more than two brain regions. In other words, these measures cannot distinguish direct and indirect connections. On the other hand, model-based methods, such as the dynamic causal modeling, can simultaneously characterize the directed connection between multiple brain regions. However, since these models use ordinary differential equations, and they are typically for low-dimensional brain networks, and also as deterministic models, they are sensitive to the model specification, data noise, and data sampling frequency. To address the limitations of existing methods for the directed connectivity, we propose a multivariate autoregressive state space model for analyzing intracranial EEG data. Let me first explain the notations. We let a d-dimensional vector yt be observed intracranial EEG measurements of d brain region. At time t, each brain region corresponds to one intracranial EEG electrode. And similarly, we let xt be the underlying neuronal state functions of the d brain regions at time t. Since intracranial EEG data have nice properties, high spatial resolution, high temporal resolution, and high signal-to-noise ratio, we propose to use a simple linear model to link YT and XT. We use a simple linear model to link region I observed intracranial EEG measurement at time t with its neuronal state function at time t. And here Ci is an annual constant, and epsilon it is the data measurement error. For the state model, we propose a first-order autoregressive model. In other words, we let region I's neuronal state function at time t depend on other brain regions' neuronal state functions at time t minus 1. The model parameter Aij represents the directed connectivity from region J to region I. And the eta it is the model error. Here is our state space model. One thing I want to point out is that our state space model is different from the standard state space model. This is because the number of brain regions and D is usually larger than 50 and sometimes can surpass 100. If we use a standard method to estimate this high-dimensional state space model, we'll likely have large variations in the model parameter Aij estimate. To solve this issue, we introduce binary indicators gamma ij's for these model parameters Aij's to distinguish non-zero directed connections from 0, 1. So with these binary indicators, we focus on identifying non-zero directed connections between many brain regions. And typically, to solve such a high-dimensional model, we can impose sparsity constraints on the binary indicator. However, simple sparsity constraints are not sufficient to produce scientifically meaningful estimates in our case. And this is because we have many network nodes, so there are enormous possible sparsity network patterns among these network nodes. And among these network patterns, most of them are not scientifically meaningful. But we want to have scientifically meaningful estimates, so we propose to incorporate existing knowledge into our model. So what is the knowledge of brain network patterns we incorporate? We found that enormous brain network studies suggested that the modular organization of brain networks, also called modularity, is tied with the two principles of the brain's functional organization, the functional specialization and the functional integration. Here is an example of modular brain network. Here are 3 modules. Regions in the same module have dense within module connections and shared the same or similar brain functions. Regions in different modules have sparse between module connections. The within module connections and between module connections ensure the functional integration of different functionally specialized brain areas. So given this prior knowledge, we believe the brain network should have a modular organization. And to impose modularity on the underlying brain network, we can impose modularity on the binary indicators of directed connection, the gamma ij. Now the challenge is, we only know the underlying brain network have a modular organization, but we do not know which regions fall into which modules. So in other words, we need to identify the module labels and identify directed connections at the same time. To address this challenge, we propose a stochastic block model motivated prior for the underlying unknown directed connection. And let me first briefly explain the stochastic block model. A stochastic block model is a generative model for the modular networks. However, it is mostly used for observed networks. For example, in a social network study, each network node represents a subject, and each network edge represents an observed relationship between 2 subjects. And this is typically what where stochastic block model is applied to. However, we are dealing a much more difficult case. because our ages are unknown, we need to simultaneously identify modules and direct the connection. So this hasn't been achieved by existing works. So instead of using a two-step approach, like we first estimate the ages and then apply the stochastic block model, we modify stochastic block model as a prior for the binary indicator, the gamma ages, and we propose to make inferences about the module labels and the directed connections within a unified Bayesian framework. Now I will explain our stochastic block model motivated prior, and I will first explain the notation. We let K with a pre-specified number of modules. For each region I, we let the Mi label its module. Mi is a k-dimensional vector with only one element being 1 and the rest being zeros. For example, Mik equals 1 indicates that region I falls into module K. We let B be a k by k matrix. For the diagonal entries, BKK denotes the prior probability of a non-zero within module directed connection between two regions in module K. And for the off-diagonal entries, BK1, K2, it denotes the prior probability of non-zero between module directed connection from a region in module K2 to another region in module K1. So here is the direction. Now let me explain our stochastic block model motivated prior. For the binary indicator of directed connections, the gamma ij conditional on module labels and the prior probability matrix are independently follow Bernoulli distribution. For example, if Mik1 equals 1 and Mjk2 equals 1, and this indicates region I falls into module K1, region J falls into module K2, then the Bernoulli probability is BK1, K2. So which is the prior probability of existing a non-zero directed connection from the module K2 to module K1. And for the module labels, we let them be IID with a multinomial distribution. For the parameter of the multinomial distribution, we let them follow Dirichlet distribution. For the prior probability matrix, we let the prior probability for a within module connection be IID with a uniform distribution truncated at a lower bound L0. And we let the prior probability for between module connections be IID with a uniform distribution truncated at an upper bound U0. And we let this L0 be much larger than U0 to reflect our prior belief that within module connections are much denser than between module connections. And we propose a Markov chain Monte Carlo method to simulate from the joint posterior distribution. So far, I introduced my model. And do you have any questions?
How do you choose K? Especially since if you're in an intracranial EEG setting, you're not getting the entire brain.
Yes.
So
before we run our Markov chain Monte Carlo, we have an initializing step using EM algorithm. And when using algorithm, we let the K be the number of brain regions. So we start from the most sparse case. Let each region falls into only one module, in a module only counting cells. We pre-run the EM algorithm to let the regions group together, and we use the output of EM algorithm to determine the number of K. And then we start our Markov Monte Carlo method.
Thank you. That's all.
Yes, thank you for asking. Any other questions?
A quick question. So
sure,
can we understand these modules as a clusters of
yes.
Okay, thanks.
Thank you. One question or criticism we have commonly received is that how can we use a model with such a simple form, a first-order model, to study the brain of such a highly complicated network system? So here I want to emphasize that we do not use our model as a comprehensive model to explain all the neuronal activities. Instead, we use our model as a working model to identify direct connections. And we detect directed connections by detecting the existence of temporal dependence among regions' neuronal activities. And this identification of the existence of temporal dependence is robust to violations of model assumption. And now let me show a simulation study to illustrate this. We purposely generate data from the dynamic causal modeling and use our model to analyze the simulated data to show the efficiency and robustness of our model. And this is an example of the dynamic causal modeling. It includes lots of ordinary differential equations. We generate data from the dynamic causal modeling for the following three major reasons. First, DCM is the most dominant model for direct connectivity in the neuroimaging field. Second, DCM is arguably a generative model of EEG data. And third, DCM is highly distinct from our model, as we can see here. So we want to show that even the underlying model is highly distinct from our model is still efficient. Now let me show the simulated network structure. Our simulated network contains three modules. Regions in dark blue, red, or green are in the same module. Gray edges represent the within module connections. Pink edges represent the between module connections. We simulate time series data based on this network structure and analyze this time series data using our method, the BSBM, and other competing methods. The multivariate autoregressive model with a L1 regularization term, partial directed coherence, spectral synchrony, graphical lasso, and transfer entropy. From these ROC curves, we found that our method achieves the largest area under curve and outperform all other competing methods. So this also demonstrates the robustness and efficiency of our model. Now, let me show the most exciting results, the real intracranial EEG data analysis results. Let me first show the example of patient 1. The left figure shows his intracranial EEG electrode placement. The right figure shows the estimated brain networks during his seizure development. Again, regions in the same color as the same module. For example, the red, dark blue, green, or yellow. Regions in light blue is in a module only contain itself. So these regions with light blue are in individual modules. The seizure onset zone of this patient is G37 and marked in diamond in this estimated brain network course. Here I want to point out that when we estimated this brain network, we did not use the information about the seizure onset zone. In other words, we treated the seizure onset zone as unknown when we estimated the brain network. And regions in the same module as the seizure onset zone are in red. We believe these regions are affected the most by seizure onset zone activity. And later I referred this red module as the seizure onset zone module. Now, let me start to show the video of the evolution of brain networks from 300 seconds before seizure on time to 300 seconds after seizure onset. Before the seizure onset, the brain networks are stable. We see that the seizure onset module is small, and we do not observe many connections of the seizure onset module. This is because before seizure onset, only a few regions are affected by the seizure onset zone activity. However, once the seizure occurs, we can observe dramatic changes. The size of the seizure onset zone module increases, and the number of connections of the seizure onset zone module increases. as the time passes, more and more regions fall into the seed-onset module, and eventually all regions fall into the seed-onset module. This indicates that the seizure spread to the entire brain of this patient. To the ends of the seizure, the brain networks return to a state even sparser than the brain network before the seizure onset. And now, let me show the second example of patient 2. Again, the seizure onset zone are in diamond in the brain network plot and are treated as unknown when we estimate them. Let me show the brain network evolution during the seizure development. Before the seizure onset zone module is small. The number of connections of the seizure onset zone is small. Again, this indicates that before seizure onset, only a few regions are affected by the seizure onset zone activity. I'm sorry. However, right after the seizure onset, the seizure onset zone size increases, and we can observe dramatic increases in the connections of the seizure onset zone module. And to the end of the seizure, the brain networks return to a state similar to the brain network before the seizure onset. Oh, sorry. So, however, unlike the previous patient, we do not observe all brain regions fall into the seizure onset module. This is because these two patients have different epilepsy types, and our method can successfully reveal the evolution of brain networks for different epilepsy types. Now I will play this video again, and this time please pay attention to the regions on the TMP electrode array and TMA electrode array. Let me start the video. During the seizure development, we find that these regions never fall into the seizure onset module and never have any connections with the seizure onset module. However, when we examine the intracranial EEG recordings, we can identify epileptic activities in this brain region. So this indicates that our method can distinguish regions directly affected by the seizure onset zone activity from the regions indirectly affected by the seizure onset zone activity. For example, we put regions directly affected by the seizure onset zone activity in the seizure onset zone module, the red module, and this cannot be achieved by other methods and visual examination. And when I explained the epilepsy at the beginning of this talk, I mentioned that one important test of epilepsy diagnosis is the seizure onset zone localization. And here I will explain the directed connectivity properties we revealed of the seizure onset zone and how we use them to identify seizure onset zone. And the left figure shows the intracranial EEG electrode placement, and the right figure shows the changes in directed connectivity of all his brain regions during the seizure development. And the red dashed vertical line indicates the seizure onset time, and this right trajectory, red curves shows the directed connectivity changes of the seizure onset zone. We found two properties. First, at the seizure onset zone has the highest changes in directed connectivity among all the brain regions. Second, before seizure onset, the seizure onset zone's directed connectivity properties are now distinguishable from other brain regions. So combining these two properties together, we develop our own new theta on-site zone localization method. And we applied our theta on-site zone, we applied our own theta on-site zone localization method to 6 patients with different epilepsy types and achieve 100% true positive rate and less than 3% false positive rate in localizing their theta on-site zone. Here, I want to point out, we emphasize that for this patient 6, he has 3 distinct theta on-site zone locations, and our method can identify all of them. This hasn't been achieved by existing method. In summary, we propose a first-order model to identify the high-dimensional directed brain network of patients with epilepsy. The proposed Bayesian framework incorporates the prior knowledge of the modular networks into the model construction. We revealed the temporal evolution of each patient's brain network from normal to abnormal epileptic state. And we uncovered unique directed connectivity properties of the theta on-site zone. And using these properties to locate theta on-site zone, we achieved very high accuracies. And this is a summary of our first model. But our first model still has limitations on the brain network study. This is because intracranial EEG is an invasive method. Intracranial EEG data are generally collected from patients with epilepsy or brain tumors. Therefore, we cannot collect intracranial EEG data from healthy subjects. And the intracranial EEG requires the activity of only a small portion of the brain. This means we cannot investigate whole brain networks using intracranial EEG data. And intracranial EEG electrode placement varies across different patients. So our analysis of intracranial EEG data can only be performed subject by subject and cannot be generalized to the population of healthy subjects. So despite the success of our method in identifying epileptic brain networks, and our analysis on intracranial EEG data still has limitations. And since we also want to investigate the whole brain networks of healthy subjects, we propose to analyze fMRI data. Let me briefly explain fMRI data. fMRI is one of the most popular neuroimaging techniques used in human brain research. This is because fMRI data provide non-invasive measurement of the entire human brain's activity, also with a high spatial resolution. And the left figure shows an example of how the fMRI data are acquired. And the right figure shows an example of the fMRI data. Typically, we can view the fMRI data as a four-dimensional time series. Since the whole brain directed networks also have modularity, and because of the functional specialization and functional integration, we extended our first Bayesian model to study many subjects' fMRI data and characterize their whole brain modular networks. We modified our stochastic block model motivated prior to accommodate the variations of brain networks across many subjects. And we combined many subjects' fMRI data to estimate the subject-specific and population mean brain network. And due to the time limitation, I won't go through the details of our second model, but I want to emphasize that the biggest challenge of this work lies in the computation, because we need to simultaneously analyze thousands of subjects' fMRI data. And I will explain how we address this challenge. First, it's computationally infeasible to directly simulate from the posterior distribution with a Markov chain Monte Carlo method. And this is because we analyze thousands of subjects' fMRI data. So to address this challenge, we propose a variational Bayesian method. Let me briefly explain the idea of variational Bayesian method. The idea is to find an analytical tractable approximation distribution of the target posterior distribution. We first assume an approximating distribution family of the target posterior, and we then find the optimal approximating distribution by determining the free parameters in this distribution family. Here, I will use our model as an example to illustrate this idea. The first formula is our target posterior. Theta is our parameter. The first part is the likelihood. The second part, p theta, is the prior. And since the number of subjects is in thousands level, we cannot directly draw MCMC simulation from it. Therefore, we propose the following approximating distribution family, Q theta, and use a regional Bayesian method to solve it. The approximating distribution family is a product of multiple independent distribution factors, Q1, Q2, Q3, Q4. And these distribution factors are typically well defined and easy to use distribution. We use these distribution factors to approximate the marginal posterior distribution of these parameters, model parameters. And the parameters in red are the free variation parameters of this distribution family, the phi IJS, phi M, phi P, phi B. And we want to determine these free parameters to find the optimal approximating distribution. And to achieve this, we propose an iterative algorithm that iteratively minimizes the following KL divergence between our approximating distribution Q and our target posterior distribution P. So in this way, we convert an MCMC simulation problem to an optimization problem and achieve the computational efficiency. Here, I will show exciting real-time format data analysis result. We analyzed about 1,000 healthy subject data from the Human Connectome project. Each subject data program consists of functional image at 1,200 time points. We divided the whole brain into 264 brain regions based on Powers atlas. And here, I want to explain more details. In the Human Connectome project, each subject has a resting state fMRI data in 4 runs collected on 2 separate days. So here, we choose 2 runs collected on separate days to form 1 fMRI data set, and use the remaining 2 runs to form another fMRI data set to demonstrate the reproducibility. So here, I will show the results based on the first fMRI data set. And these are the identified population mean whole brain directed network. Again, regions in the same color, such as green, red, or blue, are in the same module. We first compare our identified modules with existing literature, and found that our method identified modules specialized for different functions, such as the visual function, the somatomotor function, default mode function, frontal parietal function. In addition, we identify smaller modules in large systems. For example, the visual system, the default mode system. And we found that our smaller modules are consistent with the existing subdivisions of the functional system. So this indicates that our method can successfully detect the subtle functional differences between these subdivisions and reveal the hierarchical modular organization of the brain. So far, I introducing our result of the module identification. Now I will show the result of directed connection identification. Generally, the strongest directed connections are between regions in the same module, or between regions in the modules with similar function. For example, the several visual modules. However, what we are most interested are the directed connections between modules with distinct functions. And here we found a strong between module directed connections between the somatomotor area module and the auditory module. and also between the singular or peculiar type control module and the salience module. I want to say that existing methods such as functional connective methods such as ICA can also identify the strong connections between somato-motor and auditory, but they typically put them into one independent component. Here, we identified strong directed connections between these two modules, but we also separate them in different modules and suggest that these regions are related to different brain functions. Here is our identified population mean brain networks based on the second alpha data. We found that the brain networks are generally similar to the previous brain network, so which also demonstrates the reproducibility of our method. And here are the publications related to these two models. And I want to thank my PhD advisor and also my collaborators for this work. Thank you again for attending my talk. Any questions?